In this video, we'll discuss about adjuvant. What is adjuvant? When an antigen has low immunogenicity. When antigen has low immunogenicity, adjuvant are used to boost the immune response. So adjuvant used to boost the immune response. In other words, adjuvant are substances that when mixed with an antigen, this adjuvant mixed with an antigen and injected with it enhance the immunogenicity of that antigen. So adjuvant are substances that when mixed with an antigen and injected with it enhance the immunogenicity of that antigen. So immunogenicity of the antigen would be increased if use the adjuvant. Immunogenicity increased if use the adjuvant. Because sometimes antigen are not enough to boost immune response. In that case, we use adjuvant along with antigen to enhance the immunogenicity of that antigen. So in this case, sometimes antigen not enough to boost immune response. In that case, we use adjuvant along with antigen to enhance that immunogenicity of that antigen. Example of adjuvant, first one is aluminium potassium sulfate. It is also called as alum. Aluminium potassium sulfate alum. First example. When an antigen is mixed with alum, that means adjuvant, the antigen get precipitated. When we use alum is an adjuvant along with antigen, the antigen get precipitated. This one is antigen, this one is alum. So here precipitation of antigen. So that the effective time of exposure to this antigen increase. This precipitation also increase the size of antigen, thus increasing phagocytosis process. So in this precipitation result increases that size and increases phagocytosis. Because we knows if the size of antigen is more, it's large, the phagocytosis will be more easier to occur. Size of antigen more means that phagocytosis also more. So in this case, when we are applying that alum along with antigen, the time of exposure is increased because of precipitation of antigen, size is increased and phagocytosis also increased. So when we are applying this alum along with antigen, precipitation of antigen will increase, size increased and phagocytosis process also increased. Second example of adjuvant, front incomplete adjuvant. Incomplete adjuvant. In this case, the adjuvant is the oil tablet. So in this case, oil tablet is the adjuvant. When we use oil tablet as adjuvant, this oil tablet surrounded the antigen. So this is oil tablet. This one is the antigen. Here antigen adjuvant complex formation. Now this antigen surrounded by oil tablet. So antigen adjuvant complex is ready. Hence the antigen is released very slowly from the site of infections. as antigen is surrounded by adjuvant complex or oil traplet. Third one is front complete antigen. Second one is front incomplete antigen. This one front complete adjuvant. This one is one more type of antigen, that adjuvant. Front complete adjuvant is the one more type. This is same as front incomplete adjuvant. It contain heat killed mycobacteria as an additional ingredient. Here excess it have heat killed mycobacteria. Almost similar to incomplete adjuvant, where we use oil tablet, but in this case the extra things is mycobacterium, that is heat killed one. So mycobacterial, why does used here? Mycobacterial cell wall containing muramyl dipeptide, which activate macrophages, makes this adjuvant far more potent than the incomplete form. That means the front complete adjuvant is more potent than front incomplete adjuvant, because in this case we use mycobacterium. This mycobacterium cell wall, they containing muramyl dipeptide, that muramyl dipeptide agent activate macrophages. We know if macrophages activated, immunogenicity will be easier to happen. So this is all about adjuvant. Now roles of adjuvant. This one, increase number of cells involved in immune response. Immune cell counting increasing. Second one is immunogen studies. It helping for immunogen studies, more efficient processing of immunogen, then prolonging the presence of immunogen, then increasing rate of synthesis of antibodies. So increase synthesis of antibodies. So this is all about adjuvant. Thanks for watching.